Have you ever been told you have your dad's nose or your mom's eyes? That's inheritance in action. It's the process where the characteristics or traits that make you you are passed down from one generation to the next. The core of this entire process is the molecule called DNA. DNA contains all the secret instructions for building and running your body, and those instructions are packaged into small units called genes. Understanding genetics is just like decoding a super complex intergenerational instruction manual. Let's zoom way in. The blueprint of life, DNA, is a long, twisted ladder structure. When your cells get ready to divide, this massive amount of DNA coils up tightly into compact structures called chromosomes. Think of the DNA molecule as a long thread, and the chromosome as that thread perfectly spooled up and ready for transport. A gene is simply a specific tiny segment on that thread. It's the instruction for making one specific protein, which in turn often controls one specific trait, like your blood type or the curliness of your hair. If a gene is the instruction for a trait, then alleles are the different versions of that instruction. Think of it like a computer game. The eye color gene is a required feature, but the alleles are the options you can choose from: blue, brown, hazel, green. Since you get one set of chromosomes from your mother and one set from your father, you receive two alleles for every single gene. The combination of those two alleles is what ultimately determines which version of the trait you express. Before anyone even knew about DNA, a simple monk named Gregor Mendel cracked the code of inheritance. Working in his monastery garden in the mid-1800s, he meticulously crossbred thousands of pea plants. Why pea plants? Because they have very clear, discrete traits. They're either tall or short. Their seeds are either yellow or green. Mendel's simple, careful math and observation skills led him to discover the foundational rules of genetics, earning him the title, the father of genetics. Mendel's very first big discovery was the law of segregation. It states that for any trait, the two alleles you have separate into different sex cells or gametes. When you produce an egg or a sperm, that cell only gets one of the two alleles you possess. It's a 50-50 shot. When that gamete joins with a gamete from the other parent, the offspring gets a random combination of one allele from each parent. This separation and random combination is the core driver of how traits are passed down. Not all alleles are created equal. Some are dominant and some are recessive. A dominant allele, which we always represent with a capital letter, like T for tall, is the boss. If you have even one copy of the dominant allele, that trait will be expressed. The recessive allele, represented by a lowercase letter, T for short, is the quiet one. It's only expressed if the dominant allele is completely absent, meaning you have to inherit two copies of the recessive allele to see that trait. When we talk about an organism's specific allele combination, we're talking about its genotype. It's the underlying genetic code. If you have two of the exact same alleles, like TT or TT, you are homozygous for that trait. If you have two different alleles, like TT, you are heterozygous. Understanding the difference between homozygous dominant, TT, homozygous recessive, TT, and heterozygous, TT, is the first step in predicting how a trait will appear. While the genotype is the hidden code, the phenotype is the observable result. It's what you can actually see. If an organism's genotype is TT, homozygous dominant, its phenotype will be tall. If its genotype is TT, homozygous recessive, its phenotype will be short. Here's the key. If the genotype is TT, heterozygous, the dominant allele takes over, and the phenotype is still tall. Therefore, two different genotypes, TT and TT, can lead to the exact same phenotype, tall. How do scientists predict the outcome of a genetic cross? They use a simple, powerful mathematical tool called the Punnett square. It's essentially a chart that organizes all the possible combinations of alleles that an offspring could inherit. By setting up the square, we are visually representing Mendel's law of segregation and calculating the probability of each outcome. It's a quick, easy way to use data to support claims about heredity. The simplest cross to perform is the monohybrid cross, which tracks the inheritance of just one trait. Let's look at the classic example, crossing two heterozygous tall plants, TT times TT. The Punnett square shows us the possible offspring genotypes: 1 TT, 2 TT, and 1 TT. This gives us a 1 to 2 to 1 genotypic ratio. More importantly, it shows us that 3 out of 4 offspring will be tall and 1 out of 4 will be short, giving us a 3 to 1 phenotypic ratio. Genetics is all about probability. When we use a Punnett square, we're not saying exactly what will happen, but what the likelihood is. If a square shows a genotype in one of the 4 boxes, there's a 1 in 4 or 25% chance of that occurring. Think of flipping a coin. You know it's a 50-50 chance, but you can't predict the outcome of the next toss. Genetics works the same way. We calculate the genotypic probability and the phenotypic probability based on those odds. Mendel also discovered the law of independent assortment. Imagine a plant has a gene for height and a gene for flower color. This law says that the 2 genes are inherited independently. When the gametes form, the height alleles separate without being influenced by how the flower color alleles separate. This random shuffling of different traits during the formation of sperm and egg cells is one of the main reasons why siblings from the same parents can look so different from each other. To demonstrate independent assortment, we use the dihybrid cross, which tracks 2 traits at once, like seed shape and seed color. This requires a 4x4, 16-square Punnett square. While more complex, the results are powerful. If you cross 2 individuals heterozygous for both traits, e.g. AABB times AABB, the result is a distinct 9-3-3-1, which represents all the possible combinations of the 2 traits. It's a key mathematical demonstration of how much variety can be generated from 2 parents. Mendel's simple dominant recessive rules explain many traits, but nature is full of variations. We call these deviations non-Mendelian inheritance. For example, sometimes neither allele is truly dominant, leading to a blending effect. That's called incomplete dominance. Other times, both alleles are fully expressed at the same time. That's called codominance. Even in these more complicated cases, the Punnett square still works as a tool. We just have to adjust how we interpret the resulting phenotypes. In incomplete dominance, the heterozygous genotype results in a phenotype that is a mix of the 2 homozygous phenotypes. Think of mixing paint. If you cross a plant with red flowers, RR, and a plant with white flowers, WW, the offspring will have pink flowers, RW. The pink color isn't a new allele. It's the result of only a little bit of the red color being made. Not enough to be fully red. The math still works, but the visual result is a true intermediate blend. Codominance is different from incomplete dominance because both alleles are fully and equally expressed. There is no blending. A great real-life example is the roan coat color in horses or cattle, where the animal has both red hairs and white hairs interspersed. Neither color dominates, and neither is blended into pink. Instead, both are expressed simultaneously. Another classic example in humans is the AB blood type, where the body expresses both the A and A dots, protein and the B protein on the surface of the red blood cells. Some traits are considered sex-linked, meaning the genes for them are found on the sex chromosomes, typically the X chromosome. This matters because females have 2 X chromosomes, while males have 1 X and 1 Y. For males, if the one X chromosome they have carries a recessive trait, like colorblindness, they will express it. Females, however, have a second X that usually masks the recessive allele, making these traits much more common in males. When scientists track genetic traits through human families, they use a chart called a pedigree. This chart uses standardized symbols, squares for males, circles for females, and shaded shapes for affected individuals, to map out the inheritance pattern across generations. Pedigrees allow scientists to determine if a trait is dominant, recessive, or sex-linked. By analyzing past generations, they can use the same probability rules we've discussed to calculate the risk for future offspring. For instance, the chance of a person being a carrier for a disorder. So where does the variety in alleles actually come from? The ultimate source is mutation, a spontaneous change in the sequence of DNA. Most mutations are harmless, and many are even repaired by the cell. However, a mutation that occurs in a gamete can be passed to the offspring and introduce a brand new allele, creating a new trait variation. While people often think of mutations as bad, they are the essential engine of variety, ensuring that a population never becomes genetically stagnant. In the end, all of these mechanisms, Mendel's laws, non-Mendelian patterns, and mutations work together to create genetic variation. This variation, the wide array of alleles within a population, is the key ingredient for evolution. Think of it like a deck of cards. The more unique cards in the deck, the better the population can handle different challenges. When the environment changes, the organisms with the most beneficial trait variation will be the ones to survive and reproduce, a process known as natural selection.